Hi, I'm Bruce Stillman. My lab's primarily interested in how the genome duplicates not only just the DNA but also the chromatin associated with that DNA. And we are interested in the process in eukaryotic cells, nowadays using both yeast, the Saccharomyces cerevisiae, and human cells. Our primary interests are in understanding the biochemical mechanisms of how DNA replicates and how proteins are inherited from one cell generation to the next, but also how this process is controlled during the cell cycle, how it integrates to chromosome segregation, and also how this process influences cell development and also the control of genome integrity, such as what goes wrong in cancer cells. Our systems have varied over the years. Initially, we studied eukaryotic viruses that infected mammalian cells, such as SV40, and by that route, we were able to figure out the detailed biochemistry of the proteins involved in the eukaryotic DNA replication fork. More recently, we've become interested in the process of initiation of DNA replication, and through our discovery of the origin recognition complex and other proteins, we've begun to focus on how this initiation process is controlled and integrated into many diverse cellular processes. For instance, we've found that ORC is involved not only in initiation of DNA replication, but also in centrosome duplication and chromosome segregation. And more recently, we have found that one of the ORC subunits patterns the nucleus during G1 phase. And thus I've become interested in how this patterning is influenced in different cell types throughout development, from the very earliest embryonic stem cells that are pluripotent, all the way through to differentiated but proliferating cell types that express different genes. The big question is, how is patterning of DNA replication influencing the patterning of the genome for cell development? One of our also long-term goals is to understand how the process of epigenetic inheritance or inheritance of chromosome states occurs. And we've discovered proteins such as chromatin assembly factor 1 that interacts with the replication fork proteins that is involved in the inheritance of the histone proteins that are made up as part of the chromatin. And this has implications into the inheritance of gene expression states over many cell generations. And principally, we're using the yeast Saccharomyces as a system to study this. In the long run, we'd like to understand in detail how the genome is replicated completely in a very accurate way, and what goes wrong in cellular processes that lead to pathologies such as cancer.